Okay, first of all, I want to thank you all. This is such an incredible community, and thank you, John, for recruiting me into it so many years ago. Today, I want to talk about living measurement systems for engineering biology. The basic idea is to use biology to measure biology, and then by extension, to control and engineer biology. So I was really excited for this workshop. And by the way, I'm Elizabeth Strychalski. I lead the cellular engineering group at NIST, the National Institute of Standards and Technology. I was excited for this workshop because I'm hoping that it can help us understand what next best project to work on to bring minimal cells to bear on NIST's mission. So I'll introduce NIST, and then I'll give an overview of the measurement capabilities in my group, all of which we can apply and modify for minimal and synthetic cell research. And I'd very much appreciate your input on what measurement challenges you'd like to see NIST invest in this technical area. So NIST mission, we are one of the national metrological institutes in the world, and we are the Metrological Institute for the United States. And our mission is to advance measurement science, standards, and technology for economic reasons. So we're part of the Department of Commerce. And there are a number of different ways to engage with NIST, depending on what our collaborative goals might be. But I want to point out that we do more than just standards. We kind of go up this pyramid where, a lot of the minimal cell work is very fundamental, cutting edge science. So it might live down in the measurements and data part, right? What should we measure? What kind of data would be most informative for the questions we're asking? And then as we develop more and better assays, we can start to overlayer this measurement assurance part. And so there's a lot that NIST can offer even before we're developing standards. So we founded a new group at NIST with a mission to provide a foundation of good measurements to support control and rational design of biological function. Because if you could predict the function of a biological part in context and the function of many parts integrated to realize increasingly sophisticated functions, we'll be much better positioned to understand how to build safe, reliable biotechnological products fit for our desired purpose. So importantly, we'll also get a better handle on what is possible, because right now, as we know, with how rapidly biotechnology is advancing, this is a really tantalizingly open question. And I think that this community is really at the forefront of a potentially really empowering and exciting aspect of this. So I'm putting up this next slide because I wanted to just briefly flash up to you the different capabilities that I have currently in my group. And these are always evolving, but we're kind of like a lasagna where we just keep layering on new measurement capabilities. And for example, we don't actively do fluidics research as such anymore, but we have this in our back pocket if we needed to make a measurable platform, as in the case for the morphology work on SYN 3.0 and related strains. So I'm including this for your reference. But I want to briefly introduce 3 areas my group is working on to advance living measurement systems and control. But firstly, why living measurement systems to enable control? And I would argue that it's the right technology for the job, right? I mean, biology is always measuring itself all the time at every level. And I do want to mention that there are other people who are trying to insert, for example, semiconductor electronics-based sensors inside cells. And that's part of a different conversation, but one that I think might actually be really fruitful and interesting for this group. So sensors, computation, and chassis. So we're working on sensors because if you can't sense, then you can't know what your system is doing and you can't correct that function or output. In other words, it's really challenging then to implement your control strategy. Computation then allows you to process on information from multiple inputs from the environment or internal to the cell that can greatly increase your options for control, actuation, reading out measurement results, and so on. And chassis, of course, which is near and dear to the hearts of this group, we have some efforts in cell-free systems and, of course, genomically minimal cells. And what I love about this category of research is that it lets us get to the problem of what is life, how can I control it, and what are the boundaries of what it can do? And stated differently, those questions are, what is a given biological system? That's identity. What can it do? That's function. And what could it do? What are the possibilities? So first, sensors. Again, this is a slide I'm flashing up because we have this biofoundry. It's the first biofoundry at NIST called the Living Measurement Systems Foundry. And I just wanted to list off to you some of the hardware we have in this and some of the automated capabilities. We also have other automation suites that I have access to. This one's primarily for microbial work. We also have a mammalian system. There's so much I could talk about here, but I just want to let you know that one thing we're looking towards is how do we automate protocols for the minimal cell? And then what would be possible if we could do that? What kinds of questions could we start asking in this community? So far, what I like about this sensors project, and this does use that LMSF that I just showed, is it perhaps best realizes the big picture vision of engineering biology, where you want to design some function in a precise way and put it into a living system and then have it perform according to how you expect it should. So what we did here was we took a directed evolution workflow, and instead of simply measuring a handful of colonies that output at the end through a selection process, we simply take our DNA libraries, for those of you who are familiar with directed evolution, and we measure the input-output curves, that performance of every single sensor variant in our library. And this is really great for a couple of reasons, because now what we can do is we can find what we weren't looking for. We can discover novel function. In this case, for example, we were able to identify these band stop sensors that, as far as we know, don't exist in naturally evolved biological systems. The other thing we can do is we can enable precision engineering of biology by either simply choosing the sequences in silico that match our requirements, right? We can simply mine the data, or we can put that data through machine learning algorithms to start looking for design principles so that we can forward engineer and then predict the DNA sequence that would give us the sensor that we want. So imagine taking this workflow for E. coli and now repeating this or something similar to it with a minimal cell. Moving from sensors to computation, Sam Schafter in my group has led the demonstration of code transcriptional RNA strand displacement circuits. And these take the power and straightforward programmability of DNA computing and instantiate it as genetically encoded RNA circuits based on Toholt strand displacement. So now you have your circuits transcribed in situ and continuously, which makes them potentially compatible for use in vivo. And now you're just at the threshold of in situ real-time monitoring of cell state, which a lot of people are interested in for all of the reasons you might want to do this, from making products using cells to developing cells as products. And in our case, we're also looking to engineer cells into those living measurement systems I mentioned earlier. And Sam's done a really incredible job demonstrating both single layer Boolean logic and amplification circuits. He's also programmed multi-layer cascades. And these were developed along with a simple model that allows very straightforward prediction and design of circuit performance. That code is available. And now that we've sort of done the requisite pace of different combinations of Boolean logic, we're going to move on to trying to instantiate neural network-like computing in vivo. So moving on now to chassis, I know that cell-free systems is an area where some of the groups present today work in. And Jane Romansova has been leading work looking at issues of reproducibility in cell-free expression systems. You know, there are lots of reasons why you might want to take the innards of a population of cells and then get it to express your DNA template of interest rather than the DNA that's present in the cell. You might want to use that cell-free system as a platform in itself, for example, for making a product of interest because you want the functionality of biology without the system needing to be fully alive. Or you might want to use that cell-free system as an intermediate tool for testing as part of the design, build, test, learn engineering workflow for biological systems. Or of course, in this group, you might want to use cell-free systems towards creating synthetic cells. Now, reproducibility has been a challenge for these systems in research settings, for a number of reasons, and we've been focusing on protocol testing and how automation in different protocols affect the preparation of your DNA template, and how that then translates into different protein yields and variability in protein expression. And we'd also like to develop an in-house automated method for lysate preparation and encapsulation. And we expect that this method would hopefully also be compatible with any kind of cell-free system created from a minimal cell. Finally, this is the work that James Pelletier discussed earlier this morning, so I won't belabor it here. But I will say that I think the concept and workflows to achieve minimal cells may end up being, in the long run, even more impactful than this particular mycoplasmal minimal cell. I'd like to see this kind of radical genome minimization extended to other cell types. And I think I share that hope with some of the folks in this group. As we know, one of the biggest bottlenecks right now for engineering biology is how do we not just print large pieces of DNA, but then boot them up in different cellular contexts. And so I think hopefully this is something we can start to take a closer look at as a community. And I'd also like to see versions of our minimal cell that are easier to measure using readily available methods, such as a version that maybe has a larger diameter or adheres to surfaces to make fluidics and optical microscopy a lot easier, or other kinds of physical manipulation. And personally, I'd also like to explore how to make minimal cells themselves into living measurement systems to report on their inner state or their environment. Also, though, to not just use them directly, but also to teach us how to better engineer cellular life in general. So with that, my gratitude goes out to all of these people. And I encourage you to reach me at this email address if you'd like to chat about anything. We warmly invite collaboration. And we currently have open positions for technicians, postdocs, mid-career researchers, and we're in the business of making friends. And I look forward to hearing from you. Thank you so much.
Elizabeth, it would seem, I know that in mycoides, in the wild type organism, we have plasmid systems that work, and I would assume, but I don't know, that they work in the minimal cell, where you can have functioning plasmids with an a mycoplasma ORI-C that would allow you to build in biosensors and other systems that might enable some of the measurement tools that you talk about. Certainly, this could be really useful as people think about adding pathways to the cell in order to evaluate how these added metabolic systems affect metabolism in a really simple cell. So, this is something I wish Carol Artigue was still on the joining us, because she knows these systems better than I do. But I know somewhere in the building here, we're supposed to have those plasmids.
Thank you, John.
Anyone else?
I'm so interested to know if anyone has automated methods worked out for handling these mycoplasma, because I would like to take them and use them, please.
We're still, manual pipette users here, but it's something that I think, you and I have talked about, that we need to do this because there is a need for our community to be able to build lots of variants of the minimal cell at a faster pace than my lab can do at present.
There are just hugely enabling and powerful tools that we can't use with the minimal cell yet, unless and until we have these higher throughput approaches.
I certainly think it's doable. And this can be a point for discussion as we talk later on.